All right, welcome to this explainer. Today, we're diving into something absolutely mind-bending, the exact biological life cycle of a memory. Now, we aren't just talking about the abstract psychology of memory here. We're going to explore how a fleeting, temporary experience physically alters the actual biology of your brain forever. Seriously, think about that for a second. Memory isn't just software running in the background. It's literal physical hardware being built inside your head. So whether it's the profound emotion of your child being born, the simple act of learning to ride a bike, or just a sudden moment of fear, we're going to follow a single memory from the exact millisecond it's born, all the way to the night it gets stored for good. How exactly does a temporary experience become a permanent biological structure? Well, the big takeaway from today's explainer should be a fundamental shift in how you view your own mind. Every new memory you make is a physical, structural addition to your brain. To get there, our roadmap today covers 5 key areas. We'll start with the hardware of memory, then look at fat and brain plasticity, connecting memory and reward, finding the exact engram, and finally, the overnight memory shift. Okay, let's jump right into section 1, the hardware of memory and how we build proteins on demand. We're starting our journey way down at the microscopic level with a massive clue from the Kavli Prize in Neuroscience, the neuron. You probably know our brains are this vast, dense network of around 100 billion neurons. And the physical reality of all memory is basically the strengthening or fading of the incredibly tiny gaps between them, which we call synapses. But here is a huge biological challenge. Neurons are incredibly long and thin. If a memory requires new, specialized proteins to strengthen a specific synapse, waiting for those proteins to be built in the main cell body and then physically transported all the way down the axon, that takes minutes, sometimes even hours. If you are a protein, walking from the cell body to the synapse might literally take days, and by the time you arrive, that fleeting moment of learning, it's completely gone. Decades of research have revealed nature's incredibly elegant solution to this vast distance problem. Instead of sending the heavy finished proteins all the way down to the synapses, the brain just sends the instructions. It sends messenger RNA, or mRNA, directly to the synapse. The manufacturing machinery actually just waits right there at the junction. It's kind of like emailing a 3D printing file directly to a local factory, rather than manufacturing a heavy product and shipping it slowly across the entire country. This ingenious system allows trillions of brain junctions to instantly manufacture the exact proteins they need, precisely when they need them, instantly locking in that moment of learning. Which brings us nicely to section 2. Fat and brain plasticity, or how we actually make those memories stick. Because now that the cellular hardware is built exactly at the synapse, how does the brain make sure these new proteins actually stay put? Well, to answer that, we have to talk about something you might not expect: fat. Your brain is actually the fattiest organ in your entire body, clocking in at about 60% fat. It's a pretty surprising fact, but a fascinating recent study from the University of Queensland proved just how absolutely vital these lipids are for learning and keeping our memories intact over time. Researchers discovered that a specific enzyme releases a saturated fat known as myristic acid. You can think of this fat as a biological glue. It acts as a crucial chemical tag that literally hooks onto the newly synthesized proteins on the neuron and anchors them right into the cell membrane. Without this fat tagging step, the neuron simply cannot strengthen its connections, and the memory just completely slips away. So this saturated fat is the literal structural glue making your memories stick. Let's move on to section 3, connecting memory and reward, and the motivation to remember. With the memory physically built and glued into place with fat, your brain now has to decide if the experience is actually worth keeping in the long run. And this leads us to a fascinating breakthrough study from the University of Maryland. For years, scientists thought the brain handled physical location and emotional feeling entirely separately. They assumed the dorsal hippocampus, which acts kind of like a mental map of where we physically are, and the ventral hippocampus, which acts more like the heart, handling how we feel and what motivates us, operated as completely distinct parallel systems. The map on one side, the emotion on the other. But get this, using ultra-thin 3D imaging, researchers found that these two distinct pathways actually converge on the exact same individual neurons. They converge right in the nucleus accumbens, a tiny region deep in the brain that we often think of as our reward center. The synapses from both regions sit within mere thousands of a millimeter of each other. So when both fire together, they amplify each other perfectly. This beautifully links the where to go with the why it's worth going. It's exactly how your brain physically links the context of an environment with the highly rewarding or deeply fearful emotion you felt while you were there. Right, moving into section 4, finding the exact engram, or the specific cells of an experience. So we've got the protein hardware, the fat tags, and the emotional weight. But which specific cells are tasked with actually holding this memory? This leads us to the concept of an engram, which is the exact specific group of cells chosen to store a single isolated event. A groundbreaking study out of Paris tracked this down using an advanced optogenetic tool called FFL-CRE. In plain English, they used literal flashes of light to tag and control the exact active cells in mice. They looked at 4 distinct, millisecond-precise moments during a fear conditioning test. They tagged cells before a shock, during normal exploration, exactly during the physical shock itself, exactly during the fear behavior when the mouse froze, and then during normal movement without freezing. Later on, they used light to selectively turn these specific cells back on to see which ones actually held the memory of the fear. And the crucial takeaway here is that a memory isn't just stored holistically by all the brain cells active during an event. It's stored by incredibly specific, almost entirely non-overlapping, isolated populations of cells. In fact, there was only about a 1% overlap. When they reactivated the pre-shock or the normal movement cells, absolutely nothing happened. But when they reactivated the exact cells that fired during the shock or the freezing behavior, the mice instantly recalled the fear. So the memory engram is strictly reserved for the cells active during the highest moments of physical stimulus or emotional behavior. Finally, as day turns to night, we reach Section 5, the overnight memory shift and archiving the data. Once that engram is established during the day, it absolutely has to be archived for long-term storage. And this massive brain-wide shift was documented by a Chinese neurosurgery research team who recorded deep brain activity in 14 human patients. Using deep 3D brain monitoring, they found this incredible pattern. On day 1 of learning something new, your hippocampus acts sort of like a driving instructor. It sits right there in the passenger seat, closely guiding your every single move through synchronized neural bursts called ripples. But after just a single night's sleep, a quiet change of guard occurs. The information flow physically reverses. The neocortex actually takes the lead, allowing the newly learned skill or memory to become automatic and stable. Now, this is a massive revelation for your daily life. It proves that the critical consolidation of newly learned information into stable, long-term storage happens way earlier than we ever thought. It doesn't take weeks for a memory to set. The literal rewiring of your cortex begins during your very first night of sleep. Meaning, if you pull an all-nighter right after learning something important, you are actively disrupting your brain's vital data archiving process. From mRNA built instantly on demand, to fat molecules acting as structural glue, to engrams isolating our most intense emotions, and those overnight cortical shifts, we are rapidly decoding the physical reality of human thought. Before I leave you with one final provocative thought to ponder, a quick reminder, please like, share, and subscribe, and press the bell icon for more useful explainers just like this one. Also, a big thank you to our sponsor. This explainer is brought to you in part by Dr. Gangadhar Physiotherapy and Rehabilitation Center at Amirpet. If you need any physical therapy or pain relief treatments, definitely visit their clinic. You can reach them at +91 8686 105177 or check them out online at dgpc.uneweb.com. Thank you. Now think about this. The biology of your memory is already being mapped out right in front of us. If we now know the exact physical architectures, the exact cells, the fats, and the pathways that make a memory, how soon might we be able to selectively activate or even entirely choose to erase the microscopic architectures of our own identity?